If you understand, clear, no problem, or if you not understand, please ask me or ask to the key. Next, today is the our main class for the your subject. Is your subject is the protein and protein formulation. This is the second subject. My second subject in case of pharmaceutical biotechnology, in case of protein and peptide drug delivery system. So basically the protein, which is the main building blocker of our body, and protein is composed by the polymer of amino acids, and whole of the body is composed, nail to hair, composed by the protein. So structure, all structure of the body, or this is called the building blocker of the protein, and protein is a polymer of amino acid, but this protein acts as a therapeutic activity. Nowadays, this is the most important area of research area, the protein drug delivery system. Different types of protein which have a different drug delivery, different therapeutic activity, and which are formulated in the different delivery system. At present, so many protein which are used as a drugs, like the chymotrypsin, chimeral fruit available in the market, and even the vaccine, all the vaccine, even the corona vaccine, which is the protein. All vaccine are the protein in nature because all are the antigens. All are antigens in natures. So all are antigens is nothing but a protein. So they are one type of vaccine is the one of the most important protein drug delivery systems. So let us see just review of the protein, just easy level. Just you can see protein and peptides, structure of proteins, classification of proteins, stability problems. This is the most important topic, stability. We discuss on the details on the one classes, stability problems, drug delivery system, marketed formulation, stability testing, conclusion, and the difference. The proteins are the large organic compound made of the amino acid, 20 different amino acid which are composed, and formation of different types of sequence and formation of different types of proteins. Actually, without protein, no life. Without protein, the animal to human, without cannot be survive. Arranged in the linear sequence and joined together by the peptide bond. And protein is greater than 50 amino acids. And peptide, these are the short polymer of form of the linked in defined order of amino acid, and it is greater than the 50 amino acid. Protein is less than the 50 amino acids, and peptide is the protein is greater than the 50 amino acid, and peptide is less than the 50. Quite protein is higher molecule than the peptides. Peptide is the quite shorter than the protein. Actually some peptides to form a protein. Actually peptide to form some subunits, and subunit to 2 or 3 4 subunit to form a proteins. Just review of this. The structure, the fourth structure, we know a primary structure of amino acid. This is the linear structure. Secondary structure, this is the regularly depending on the local structure stabilized by the hydrogen bond, and primary structure which is stabilized by the disulfide linkage and peptide linkage. And tertiary structure, these are the three-dimensional structure. An example is the all enzymes which are tertiary in structure, and they are stabilized by the peptide, disulfide plus hydrogen bond. Quaternary, same. These are the four peptides or structure formed by the several protein molecules, or like polypeptides, generally get at least 4 peptide chain, and these are stabilized by the peptide, disulfide, and hydrogen, and other hydrogen bonding, like other electrostatic force, hydrophobic interaction, van der Waals force to stabilize the quaternary structure of protein. So these are the graphical representation of primary structure of protein. You can see it just is the amino acid which are linear in structure. One is the amino group, another is the carboxyl, or vice versa. Here is the carboxyl group and here is the amino group, or C terminal to N terminal, N terminal to C terminals. Just I read fast because it is, I think it will read in the B form level. Another secondary structure, generally you know the two type structure which are alpha, another is the beta plated sheet, alpha helix and beta plated sheet. These are the helical structure, right handed or left handed helical structure, which is basically composed by the peptide bond and or disulfide, but which are joined. This helix structure, which is helix, is formed and stable by the hydrogen bonding. H, what is hydrogen bonding? Hydrogen with oxygen or other electronegative compound. These are to form a helical structure, and it will be discovered first. That's why it is alpha, and then the sheet like structure will discovered. That's why it is called the plated sheet. But the second, that's why it is called the beta plated sheet. There is no other issue. Only it was discovered first. That's why it is alpha. Then it was second. That's why it is called beta. And just like it is a sheet, asbestos like sheet. That's why it is called beta plated sheet. And just like a one sheet, just like a one chain, and the one chain, another is the hydrogen bonding between these two chain to form a sheet. Another is the tertiary structure, generally 3 polypeptide chain or primary structure, which is composed to form a, that is tertiary structure. All enzymes are tertiary in nature, and quaternary structure, hemoglobin is the example with the alpha 2, beta 4 subunit. That is called the quaternary structure. And the protein structure, like lactate dehydrogenase, it is the enzymes in the different structure, immunoglobulin fold, beta fold, and hemoglobin B chain with alpha. And the types of protein and the peptides, depending upon the number of amino acid, they are classified in the following groups, basically depend on the number of amino acid as well as the sequence of the amino acid. One is the polypeptides, number 2 oligopeptides, and a short poly, then oligo, then fibrous protein, globular protein, and oligomeric protein. According to their biological role, they will be classified into the different types of protein which are acts as a different action, actually whole of the body. The first one is the enzymes, because it catalyzes all the type of reactions and help for the metabolism. Say for example G6PD, glucose 6-phosphate dehydrogenase. Transport protein like hemoglobin of erythromycin to carry the oxygen. Contractile or motile protein, actin or myosin, which you know it, the actin and myosin light chain kinase. And structural protein collagen in bond, and defense protein in the immunoglobulins or the antibody, which are the defense protein, IgE, IgG, IgE immunoglobulin, IgM, IgG, 5 types of immunoglobulin. And regular protein, insulin, regulatory protein, because it regulates the blood sugar level. That's why it is called the regulatory protein. Insulin, which is secreted from the islets of beta cell of islets of Langerhans protein, pancreas. Nutrient and storage protein like ovum, albumin. These are the nutrient or storage which is stored in the body, as well as the which acts as a nutrient, that means having some food value or energy, giving some energy. Why protein and peptide used as a drugs? The protein and peptide are very important in the biological cell. You know, just the before slides. And lack of protein and peptide causes some diseases like diabetes mellitus. And diabetes mellitus is caused due to the lack of protein like insulin. So insulin is the main culprit for the causing of diabetes mellitus. And this insulin, which is composed by the protein. So in that case, if you replace this protein or insulin, then the diabetes will be overcome. So here insulin as a hormone, but when this insulin is a formulated form of, and which is administered into the body, which acts same function to regulate the blood sugar level, that insulin, it is called the insulin as a drug or protein drug. And if it is administered in the different way, generally still now it is administered in the subcutaneous route or parenteral route, not in the oral route. But it is a protein in nature, chemical in protein in nature, which are administered to regulate the blood sugar level. That's why insulin is a protein drug. Do you understand what is the difference between the protein and protein drug? Because দেখো প্রোটিন অ্যান্ড পেপটাইড ইজ এ ড্রাগ, এটা অ্যাকচুয়ালি প্রোটিনই, actually protein their biological value to regulate the blood sugar level when it is secreted from the beta cell of islets of Langerhans or pancreas. But when the lack of insulin in the body, then it is prepared outside the cell or laboratory or in the manufacturing unit, then it is used as a drug. Key drug, which type of drug? The protein drugs. Now nowadays the rDNA technology, basically the insulin production, rDNA technology and the hybridoma technology also require the protein and peptide based pharmaceuticals. And for your kind information, insulin is the first drug which is prepared by the rDNA technology, recombinant DNA technology, and insulin is the first drug because it is the most huge demandable drug nowadays, because most of the people are suffering from diabetes mellitus. But how it will be collect the insulin? That's why the a biotechnology is developed in the year 1948, and first drug is the insulin by recombinant DNA technology. I think you can more elaborate discuss in the rDNA technology by the master in the biotechnology subject. Do not I in that case, which protein, how it will be isolated, and where, and how to treat it of the different types of disease, and how where it will be utilized, that will our main target. Protein and protein formulation. You know what is protein, what are the chemical stability, what are the function of the body, and how to formulate the protein. Protein engineering, these are the main motto of this subject. What are the advantage? Like erythropoietin, it is the another protein which is coming from the erythrocyte, used for the production of the RBC, tissue plasminogen activator. It is also another protein for use for the heart attack or in case of shock, this plasminogen to prevent the coagulation. And the oxytocin, which you know it is a hormone, it is a peptide hormones which maintain the labor pain, because the oxytocin to maintain the labor pain for childbirth. And bradykinin, it is also protein, increase the peripheral circulation. And stomatostatin, which decrease the bleeding of the gastric ulcer. And gonadotropin, which induce the ovulation. Insulin, which maintain the blood sugar level. So these are all are protein, left hand side all are the protein, and which are the different biological value, play the biological role. But when it is formulated in the, when it is the deficiency, which are formulated in the laboratory or in the production manufacturing unit, then it is used as a protein drugs. But how it will be formulated, then discuss later. For transport or storage of the small molecules, coordinate the motion via muscle contraction and mechanical support from the fibrous protein, generation, transmission of the nerve impulse, enzymatic catalysis, immunoprotection through the antibody, and control the growth and differentiation via hormone. All over the body, which is the main function is the growth, differentiation, and this hormone, protein like in nature, or that is called the peptide hormone. All are not peptide hormone, but these growth factor, which are all are peptide hormones. So enzymatic catalysis, all the reaction will be done by the enzyme, but this enzyme is chemically is the protein. So these are very important. And the generation, transmission, nerve impulse, this is also protein in nature, mechanical support of the fibrous protein. So protein is a huge role. Actually no protein bollam, no protein no life, the huge role. But when it is formulated, which are designed as a drug, that is called the protein drug. And how it is delivered, and what are the disadvantage of the protein and peptide drugs? A very large and unstable molecule, because protein when you administer orally in presence of the hydrochloric acid or in the pepsin which is present in the stomach, it will be digested. So there is a great problem in case of formulation of protein, because if you administer, it will be hydrolyzed. So stability of the protein is very important. Next class here, the stability of protein, how to stabilize the protein. Next, another disadvantage to formulation of the protein, structure is held together by the weak and non-covalent force. Weak non-covalent force, very weak, non-covalent, easily break down, not rigid in structure. to to how to solve the problem or how to minimize the problem if you used as a protein for formulation of the drug, easily destroy by relatively mild storage condition and the gastric juice, এটা main factor. তাহলে কি করতে হবে? Either it will be coated something or either a bypass something or directly inject into the body, otherwise it will be destroyed, otherwise it will be destroyed. Next, hard to obtain in the large quantities. It's very difficult. How to large quantity, there are insulin দরকার, huge quantity, how to বা oxytocin is দরকার huge amount, how this is very hard and toughest job to collect the large number of or a large amount of protein. These are the main advantage. যে প্রোডাকশন করে প্রোটিন তৈরি করবে, যেরকম ড্রাগে তোমরা সিনথেসিস করলে প্যারাসিটামল ধরো 1 টন দরকার, সিনথেসিস করলে হয়ে গেল, এবার 1 টন অ্যালবুমিন দরকার, তার মানে সেরকম অ্যামাইনো অ্যাসিড দরকার, তো দেয়ার ইজ এ ডিফিকাল্ট, very very difficult to collect the large amount of protein in the manufacturing unit. Problems with the proteins in the elimination by the B cell or by the T cell, beta lymphocyte or T lymphocyte. Another is the main problem, the proteolysis by the endo or exopeptidase. When you take the protein drug, they are endo or exopolypeptide to digest them, and small protein filtered out by the kidney. There is another problem. Suppose that you create a protein which are for therapeutic value, but this protein filtered by the kidney because it is smaller in size. Unwanted allergy reaction. Obviously, you administer either orally or by parenterally. If a protein that is another foreign particle, it is antigens, it will be produce some chances of allergic reaction and maybe develop, and sometimes maybe toxicity, sometimes maybe delayed hypertension, sometimes maybe immediate hypertension, and maybe death may be occur. So be careful before the administration, before the formulation, that type of safety study must be done. And there is a loss due to the insolubility and the absorption, because protein water soluble substance, loss due to insolubility. Some proteins are insoluble, but some proteins are soluble or due to adsorption problem জন্য or loss হতে পারে প্রোটিনের ক্ষেত্রে concentration. Next, the stability problem and the cause. First, the physical stability. Physical stability involves in the transformation in the secondary, tertiary or quaternary structure of molecule, because primary structure is the most lowest structure. Their higher version or higher stage is the secondary, then tertiary, then quaternary. But when it is administered, suppose the quaternary structure of protein, in presence of proteolytic enzymes or some other issue like heat, pH, that destroy the physical property of the particular proteins. And one of the most important mechanism by which protein can be destroyed by the physical property of the protein, that is denaturation. Any non-proteolytic modification of the unique structure of the native protein that are affected definite changes in the physical, chemical and the biological property. No proteolytic reaction will be done. Suppose a temperature, small increase the temperature, the protein structure will be changed, and they affect in case of physical, in case of their chemical property and also their biological property. Suppose some protein have biological property having some food storage property, but after denaturation they are having lost the their biological activity or the storage or food value will be lost, or sometimes their chemical property will lost, so their chemical nature will be lost. Suppose you prepared insulin, but insulin after denaturation they do not regulate the blood sugar level, or their chemical nature will be lost, or the physical property. So there is a most important problem if you prepare a protein drug, and denaturation is one of the most important fact parameter that can be change the physical, chemical and biological property of the protein. Peptides and proteins are compromised of the both polar amino acids and the non-polar amino acids, and the factors favors the denatures. Which factors, that will be avoid that factors to aggravate to favors the denaturation. So your duty to avoid this denatures, and so avoid these factors. Which factors? When the solvent changes from a aqueous to organic solvent or to a mixed solvent, they never use. Suppose the formulation, the solvent which are water, but you change for better solubility organic solvent, there is a chance of denaturation. Or a mixture of solvent, organic solvent, aqueous solvent or organic solvent, there is a chance of denaturation. So first is the denature is the main culprit, main factor to stability problem, and this denature can be aggravated by these factors. Another is a pH. You know, because each and every amino acid having the isoelectric pH, and also protein have a definite pH, and at that pH they are very much stable. But when you change this pH, their stability problem will be arises. So how to stabilize their pH? Obviously you use some buffer system to do not change the pH. If the pH will be changed, the protein will be denatured, and the denatured will be done, then the physical, chemical, biological property will be lost. So be take care, do not change the pH. তার জন্য alternative ব্যবস্থা, alternation, the ionization of the carboxylic acid and amino acid, and thereby changes carried out the molecules. Another factors which aggravate the denaturation, alteration the ionic strength. That means amino acid, protein composed by the amino acid composed by different ions, and this combined ionic state is called the ionic strength. That will be main factor to denature the. So when a formulation will be done, you must be maintain the optimum temperature. Do not increase the temperature. Otherwise there is a chance of denaturation of protein activity of the formulated protein. Denature may be reversible or sometime may be irreversible. If reversible, that is no problem, but if it is irreversible, there is a problem. After the production will be over, then the drug will not be there, having no therapeutic activity. Denaturation may lead to increase or decrease in the solubility and alteration of the surface tension, loss of crystallization ability, change in the constituent group of the reactivity, molecular profile, molecularity of the enzymes degradation, and loss of alteration of the antigenicity and loss of specific biological activity. In case of denaturation, there is huge problems. So be take care, there is no chance of denaturation, because if the denaturation, solubility problem will become, their surface tension will be changes, their crystallization ability changes because they are more polymorphism, or change in the constitution group relative and molecular profile. So be take care, be careful, no denaturation occurred during the formulation. Or some denatured agent category like polar or protic chemicals mechanism disturb the H bond, example urea, guanidine, HCl, alcohol, acetic acid, জানো প্রোটিনের সঙ্গে এইসব দিলে denature হয়ে যাবে. Another is the surfactant. Generally in case of manufacturing unit, surfactant is used. Hydrophobic disruption and change in the group the separation, and sodium dodecyl sulfate, polyethylene glycol, and ammonium chloride. These are the surfactant which change the protein denaturation or help for the denaturation. So be take care, this type of surfactant never mixed with the protein formulation. Methods for the prevention of denaturation. How to prevent the denaturation? Denaturation protein is restored or removed or denatured. The maintaining the pH, obviously, that first, maintaining the ionic strength and maintaining the temperature. 3 parameter you can stabilize use some buffer system or by ionic strength or temperature, fixed regulation temperature. There is a less chance of denaturation. The adsorption, peptide and protein are amphiphilic in nature, hence they are tend to be absorption and interfere with such a air, water and air solid. An example is the insulin. Peptides and protein in the amphiphilic, both hydrophilic, lipophilic, and hence they are tends to absorb interface of the air, water, and conformation and rearrangement leading to denaturation can be induced by their interfacial adsorption. And after adsorption, they form some short range of bond, van der Waals force, hydrophobic, electrostatic, hydrogen, ion pair bonds, and with the surface resulting into the further denaturation of polypeptides moieties. So absorption, another is the main factor for destroying or instability or instability of the protein. Adsorption of peptides and proteins interfere with the rapid but the rate of conformational changes are relatively slower. On absorption, there may be a loss or change in the biological activity as the molecular is rearranged. If the peptide protein enters the absorption at interface, there may be a reduction in the concentration of the drug availability to elicit, that means increase its function. Such loss of proteinaceous drugs may occur during the purification, formulation, storage and other delivery. And method of prevention of absorption. What type of method or type of measures should be taken to prevent the adsorption? On insulin, adsorption may be minimized by adding 0.1 to 1% albumin. And insulin with albumin 0.1%, adsorption will be prevented. Excess agitation should prevent during production. The handscape within the confines with the container should be small. Use of surfactant to reduce the adsorption. But be take care, this surfactant reduce the adsorption, but surfactant increase the denaturation. So what type of surfactant and what concentration, that will be maintained. A smooth glass wall best to reduce the adsorption or precipitation. This container, the manufacturing unit must be smooth glass container, that will be better for prevent the adsorption. Next, aggregation and precipitation. It is another stability problem. The denatured unfold protein may be rearranged in such a manner that hydrophobic amino acid residue of various molecules associated together to form a aggregation. If the aggregation is on a macroscopic scale, precipitation occurred. Interfacial absorption may be followed aggregation and precipitation. The extended true of aggregation and precipitation occurred. Relatively hydrophobicity on surface in contact with the polypeptides and the protein solution cause this presence of large air-water interface generally the accelerate this process, and presence of the large headspace within the confines of the container also accelerate the course of precipitation. Insulin from a fine divided particles precipitate on the wall of the container and referred to the frosting, and the presence of large air-water interface generally accelerate this process. To prevent the aggregation and precipitation, organic solvents such as 10 to 15% propylene glycol can suppress the formulation of peptide. Another is the related to denaturation. Excess agitation should prevent that during production. The head space within the confinement of the container should be small. There is a less chance of agitation. The ionic strength, solvent composition, solvent dielectric profile, and pH that carefully controlled at very step in production. Used for the surfactant to reduce the aggregation. And it's a chemical instability. Chemical instability. These are the physical, denaturation, coagulation, agitation, and these are the chemical instability. Up to this, do you understand? These are the physical instability of protein. Actually, this protein and protein formulation, no standard book available still now. মানে সেইরকম কোনো বই নেই in case of the molecular cell biology. There is a specific book. So only some journals, and these are the material I can supply it, and from this material you can study, and also some journal also I provided you from that will help for kind of, because no such book available in the market. মানে মার্কেট না, মানে সেরকম কোনো বই নেই এই মুহূর্তে। আন্ডারস্ট্যান্ড? ফলে এগুলোই যে দেবো, এগুলোই দিস ইজ ইউর মেটেরিয়াল, আর কিছু জার্নাল পেপারস দেওয়া হবে, সেগুলো থেকেই পড়তে হবে। ওকে, তো আজকে এই পর্যন্ত থাক, কারণ শরীরটা আমার খুব ভালো না। ওকে
ওকে স্যার, ওকে স্যার।
মানে আমি প্রচন্ড জ্বর থেকে উঠেছি, মানে জাস্ট পরীক্ষা আছে।